about secondary RNA recognition motif RRM2 of TDP-43 protein and how it behaves under conditions of oxidative stress, what happens when it's subjected to oxidative stress. Oxidative stress could be described as high concentration of reactive oxygen species, such as superoxide radical, nitroxide R radical, and some others. And usually they appear when cell ages and when there is a disbalance in redox regulating systems occurs. And also they present in healthy cells in normal chemical milieu. These species can produce oxidative damage to proteins and other molecules in cells. And for proteins, main target of this species is cysteines, cysteine residues, and they have TO group SH, and this group could be oxidized to form disulfide bond. And when we add some reducing agent to our system, we can reduce this bond to free thiol group. It's clear that such bonding can introduce disulfide bonds both between different protein molecules, it's intermolecular bonds and intramolecular bonds. The usual case, so I will just tell about our systems. Usually such bonding stabilizes systems, but if this bonding is not native, it can produce very strong effect on structural integrity and functions of proteins. And for example, for TDP-43 protein, the disulfide linked aggregates are formed in neurons and glial cells. In patients, in brains of patients who suffer from amyotrophic lateral sclerosis and frontotemporal lobar degeneration. These diseases are widespread and important. Among with full-length TDP-43 in aggregates were found C-terminal fragments, and these C-terminal fragments are the products of cleavage of this full-length TDP-43 protein near the RRM2 domain. Therefore, we will focus on this domain. This domain has two cysteines, namely C198 and C244. One of them has a side chain exposed to the solvent, and it can easily form disulfide bond. And the second one has side chain buried inside, and it likely not form disulfide bonds with other protein molecules. And we used a set of very broad physicochemical and biochemical techniques to study the system, and we obtained this picture, and I will try to explain it in the beginning, and I will provide some evidences that will confirm this picture. Firstly, in normal reducing conditions, protein is in monomeric form. When we introduce oxidative stress, it forms disulfide bonded dimer. We have these solvent exposed side chains. This protein becomes destabilized. We observe that equilibrium between folded and unfolded state shifts towards the unfolded species. These unfolded species form aggregates, and these aggregates remain soluble. After we add some reducing agent like dithiothriatol, monomers could be released from these aggregates, but still some part of protein remain in aggregate form. And I will show you the results of some experiments providing the evidence of such picture. First experiment is SDS polyacrylamide gel electrophoresis. This experiment basically based on the migration of protein in the gel. The protein is denatured by SDS, and the velocity of migration depends on the molecular weight. And in this way we can clarify the oligomerization status of RRM2. Here we performed the experiment for 3 samples. The control one, the sample that was oxidized by hydrogen peroxide for 2 hours, and the reduced sample that was oxidized for 2 hours and then reduced by dithiothriatol for 3 more hours. And we prepared samples for wild type protein in which both cysteines are present, and for C198S mutant, where we replaced one of cysteines by serine. And for wild type, control sample contain monomers, for mutant as well. For oxidized sample of wild type, we have we observed the formation of dimer and different oligomers. For C198S mutant, we observed the formation of dimers only because it has only one cysteine, and the cross-linking is not possible in this case. Only dimers could form. And this is the first evidence that we have some oligomerization after oxidative stress is introduced in our system. The second experiment is the NMR experiment. We recorded HSQC spectra for control and oxidized sample. And we found that for control sample we have a nice HSQC spectrum with very well dispersed peaks. And when we recorded spectrum for oxidized sample, we found that intensity dropped to 60% from initial one, and it means that some signals disappear. We were looking for signals broadening in order to find these dimers or oligomers, but we didn't find any of them. It means that 60% of protein disappears, and it cannot happen because the protein didn't precipitate it, and it's still soluble, and there are no flakes in our sample. In order to find what we observe in our system, we measured N15 relaxation and found that both control and oxidized sample contain monomers of protein. And based on this data, we concluded that 60% of our protein that is not visible, that is NMR silent, is in aggregate form. So, but when we oxidized our sample further, we found that all peaks disappear. And after that, some new peaks appear that were not present in sample before. And these peaks were attributed to flexible tails of aggregates of protein that is an aggregate form. For example, this one, it migrates with the whole aggregate and diffuses slowly, but this tail is flexible enough to introduce dynamics and produce rather slow relaxation, and therefore we observe rather sharp peaks here. These peaks were used to characterize diffusion of these particles by means of pulsed field gradient NMR, and also we used dynamic light scattering to get the size of particles in our system. So, in control sample, we assumed that we have monodisperse distribution of particles. It's the monomers, and we measured the sizes and obtained the value of 1.7 nanometers. This value is consistent with the size of NMR and X-ray structures plus the hydration shell. And in oxidized sample, we have a mixture of monomers and aggregates. In DLS, we have to fit the experimental data by big exponential distribution, and we have to introduce some weight of monomers, weight of aggregates, and characteristic sizes of both particles. And we measured the weights from the NMR experiment, because the portion of NMR signal is exactly the portion of monomers in our system. And the NMR silent part is the aggregates. And we, therefore, in this way, we obtain the weight of monomers and weights of aggregates. Here on this figure, you can see how weight of monomers changes with time when we oxidize our sample. It drops from 100% to about 10% in 24 hours. And when we reduce our sample, it goes up to 60 or 70%. And then we performed fitting of our data, of DLS data, using these weights and using the knowledge about the size of monomer, and we obtained the size of aggregate particles. And here you can see that aggregate particles after 24 hours has a size of about 10 nanometers, and in reduced sample we still have aggregates, and they have size of about 5 nanometers. And also we measured NMR diffusion on this oxidized sample that was oxidized for 24 hours, and using fitting of the data with standard statistical Tanner equation, we found diffusion coefficients for these particles, and we found that for oxidized sample we see a slower decay of intensity than for control and reduced sample. It means that oxidized sample contains these particles that have a slower diffusion, and it's consistent with our previous findings. And here I want to mention that for control and reduced sample, we can see that the diffusion is the same, but from DLS we have the different result. And it's also consistent because for reduced sample we measured the diffusion for the peaks that assigned to monomers. We don't see the peaks from aggregates in reduced sample. And after that, we compared the results of dynamic light scattering, impulsive field gradient, and NMR. And here we have a rather consistent picture. So, for example, we have diffusion coefficients of aggregates very similar between NMR and DLS. One of interesting results here is we tried to estimate the amount of monomers in aggregate particles, and from DLS and NMR data so we estimated the distribution of these aggregate particles, and from NMR we have 15 particles, and from DLS we have 40, and it means that the reality is somewhere in between. So aggregate particles contain unfolded species of RRM2, and in order to find the structure, to characterize the structural stability of RRM2 under oxidative conditions, we used HD exchange experiment. The idea of this experiment is that we dissolve lyophilized protein into deuterated solvent and amide protons of protein substituted by deuterium. And this process depends on the structural stability of protein. For those regions of protein, for example, for these loops, we have amide protons exposed to the solvent, and they could be easily exchanged with solvent and substituted by deuterium. Therefore, the exchange is really fast. For structured regions, these protons are trapped in hydrogen bonds that stabilize secondary structure, and we have slow exchange here. So let's look at our experiment. It was designed in a way that we dissolve our protein and record a reference spectrum. We add hydrogen peroxide, record HSQC spectra for 2 hours. After that, we add the reducing agent and record HSQC spectra for 24 more hours. Here on these figures, you can see the decay of intensity that is associated with substitution of amide protons with deuterium. Because of this substitution, we lose the intensity. For control sample, I forgot to say that for control sample we didn't add peroxide. Instead, we added the same volume of water to the sample. And for control sample, you can see that the intensity drops. This is a normalized intensity. It drops exponentially, and it's a normal process of deuterium exchange. But in case of oxidized sample, during the oxidation period, this first 2 hours, we observe a steep decay for intensity. And it is explained by the fact that protein is structurally destabilized. These hydrogen bonds disrupt, and deuterium can penetrate in the protein structure. And this happens not for some residues, it happens for all residues of protein, and it means that this process is cooperative, and it means that full protein unfolds, and it recruited to aggregate particles. So this is evidence of protein destabilization under conditions of oxidative stress. Another biochemical experiment is trypsinolysis. Trypsin is a protease that can cleave protein in sequence after arginines and lysines, and it can cleave only unfolded or disordered regions. But if protein is folded, it's global, it cannot fold, it cannot cleave it, because the recognition sites are not accessible by this protease. And so here is a figure of represents the photo of gel, where we analyze the results of trypsinolysis. First lane shows a control sample with monomers, second oxidized sample with oligomers and dimers, and third lane shows the results of cleavage for control sample. Here we can see that it's similar to lane 1, and it means that control sample wasn't cleaved. It means that if our sample is under reducing conditions, it's structurally stable and trypsin cannot cleave it. But if we add trypsin to the oxidized sample, it cleaves everything. It cleaves dimers, it cleaves oligomers, and it cleaves even monomers. And it means that monomers that should remain structurally stable somehow recruited to aggregates, and they become unfolded and susceptible to proteolytic digestion. NMR provides more information about results of trypsin cleavage. For example, for control sample, we found that actually protein is cleaved, but it's cleaved at the C-terminus where we have some flexible tail. It could be easily accessed by trypsin and cleaved. But for oxidized sample, we found that spectrum changes completely from spectrum that correspond to folded protein to the set of unfolded peptides. So, to sum up, I will show you such a picture that I have in mind after I analyze all the data of this experiment. So we have in a normal sample the dynamic equilibrium between the folded protein and unfolded. But under reducing conditions, in case of RRM2, we have the equilibrium shifted to the folded state. And we have almost all protein in the folded state. And therefore, during time, we have stable native state. But when we introduce oxidizing conditions, we have the intermolecular disulfide bond formed. And we have change in kinetics of reactions responsible for folding and unfolding. And therefore, unfolding state become more stabilized. And after time, we have deplete of folded state pool and replenish of unfolded state pool. And after time, this unfolded species can form aggregates and be cleaved. So conclusions are such that we studied the oxidative stress from very, very different points of view, and we obtained, I think, first structural information about how oxidative stress changes the protein environment, and we obtained the broad picture, very detailed picture on how this oxidative stress can lead to consequences that are observed in neurodegenerative diseases. I want to thank funding and research part of St. Petersburg University and you for your attention.
Thank you very much. Any questions for Sebastian? Please. You mentioned the cross-relaxation between chemical shift anisotropy and dipole.
Yep.
How do you determine this complicated quantity?
Okay, this is a special pulse sequence in which we measure the ratio of intensities of upfield and downfield component of doublet. We do not apply the decoupling, and the components of doublet they relax with different rates. And this difference could be extracted, and in this way we obtain this transverse cross-correlation rate.
And did you change the external DC magnetic field in order to see the contribution of
No, it's in static field.
It means it is something like an estimation.
Actually, it's rather accurate way to measure this quantity.
Well done. Any more questions?
What kind of students are you?
I'm a PhD student. PhD student.
PhD?
PhD student.
PhD. Да, PhD. Yes. Yes. Да, PhD. Yes. Thank you. Так я и понял.
Yeah. You have talked about some NMR signals as in HSQC disappeared and became NMR silence. Why this? Why?
Okay, so if we measure HSQC spectrum of small protein, we have a lot of dynamics, and therefore relaxation of so the NMR relaxation is rather slow, and we can record the HSQC. But when we will look at bigger proteins, the relaxation will be faster, and during this pulse sequence, the relaxation will take place, and the signal intensity will drop down. And actually, this is because of, for example, if we will look at R2 values are high for bigger proteins, and effectively the lines are broad. But when we have aggregates, we have really huge proteins, and therefore the R2 is really has a high value, and therefore effectively peaks are broadened in a way that we cannot observe them.
Yeah, thank you.
The last question.
Do you measure UV spectra of your protein before and after aggregation?
Yes, we do.
And is it change?
Not actually. Probably we can say that some. So, for example, this sulfide bond should contribute to this UV spectrum, because in control sample we do not have it, but in oxidized we have. But this contribution is not that important on the scale of overall UV spectrum.
Okay.
Thank you very much. Well, I certainly have the discussion. The next speaker